Welcome, everyone. Thank you so much for joining us today. My name is Lauren Minier, and I am a Senior Program Associate at AAAS in the Inclusive STEM Ecosystems for Equity and Diversity, or ICE program. We are excited to be hosting today's webinar titled Engaging Students in Course-Based Undergraduate Research Experiences. And we are excited to hear from our presenters today, Dr. Allan Feldman and Dr. Lina Dahlberg. Before we get started with today's workshop, I have a few housekeeping notes. This presentation is being recorded, and the recording will be posted in the coming days on our website. We also have the slides and a handout for today's workshop available on our website as well. We will be going into breakout room discussions after the presentations, and those breakouts will not be recorded. But the full recording will be available later this week. Additionally, we have closed captioning for this workshop. You can download and view the closed captioning and full transcript by clicking the CC button at the bottom of your screen. And before we turn it over to our presenters, I wanted to provide a little background information about AAAS and our IUSE initiative. So the American Association for the Advancement of Science was founded in 1848 and is an international nonprofit organization dedicated to advancing science, engineering, and innovation with the specific goals of strengthening and diversifying the science and technology workforce and fostering education in science and technology for everyone. With more than 120,000 members in more than 91 countries, AAAS is the world's largest multidisciplinary science society and a leading publisher of cutting-edge research through the Science family of journals. You can learn more about AAAS at AAAS.org. And for those of you who are new to the AAAS-IUSE initiative, IUSE, which stands for Improving Undergraduate STEM Education, this initiative seeks to support faculty, students, and the greater undergraduate STEM education community by disseminating research and knowledge about STEM teaching, learning, equity, and institutional transformation. We invite you to learn more about the initiative on our website, and we hope that you'll join us as future contributors. You can also follow us on social media on Twitter and LinkedIn to stay up to date on our latest events, blogs, or to share what you've learned at today's workshop. So now, without further ado, I'd like to turn it over to our first presenter. I'd like to welcome Dr. Allan Feldman, who is a professor of science education at the University of South Florida. Allan, over to you.
Hello, everyone. Very happy to be here. I'm joined with, joining me is Rita Ortiz, who's one of my doctoral students at the University of South Florida, who's been working with me on the research we've done on course-based undergraduate research experiences. So a little bit of an overview of what I'm going to be talking about is a model of how people learn to do research that I developed with my colleagues at the University of Massachusetts when I was studying an interdisciplinary research project on the natural remediation of acid mine drainage. And then a model, a course model that I developed that I had a great participation with from the STEM faculty at the University of South Florida. I thought it was going to be a great thing to try out. I didn't get the funding from IUSE, and then the pandemic started, so we didn't have a chance to try that out. But then we did a pilot study within a existing science education course. And I'll talk a little bit about that and some of the preliminary data, preliminary results that we found. So I guess I'll just get started with that. All right, so this model for learning how to do science and research group came out of, like I said, the study of a group of people working on acid mine drainage. And basically the model is, as we probably all know, that learning to do research takes place in the STEM disciplines and apprenticeships. The apprenticeships take place in research groups. The students have different roles in those groups. They can be in the role of novice researcher, proficient technician, or knowledge producer, and that's a continuum that's not compartmentalized. And then the members of the research group, which includes the professor and other students, help facilitate the development of the students along the continuum of roles. So just some quick information about each of those different parts of the model. So to engage in an apprenticeship, if you're familiar with the work, important work of Lev and Wenger, is to engage in legitimate peripheral participation in a community of practice. So what that means is that what the apprentice is doing is legitimate work. Washing glassware is not legitimate participation in the research group. Peripheral means that the student or any apprentice starts out on the sort of towards the edges of the research experience. And then as they gain expertise, they move more and more towards the center and become more important for that. So characteristics of apprenticeships. Well, important thing is that the nature of learning and the practice of work are indistinguishable. Apprentices learn by doing. An important part of an apprenticeship is that the master, in our case, it would be probably the professor who runs the research group, partitions the tasks that the apprentice is going to do in a way so that as they engage in those tasks, they are learning. So it's a scaffolded process. And then the way that you demonstrate, the way in which an apprentice demonstrates what it is that they know is they do it. So by successfully doing what those tasks are, it demonstrates that they're capable of that. And then they can move on to more complex or more challenging tasks. So then a research group is defined as a lead researcher, a group of students, possibly a postdoc, and possibly technical staff. The group is defined by the area of study, and most importantly, the lead researcher, who is often a professor at a research university, but it could be in industry, could be in a nonprofit. And then one of the things that we found in this research was that we could talk about configurations of research groups is in two ways, either tightly structured and loosely structured. Now this diagram, it's a little complicated, but not as complicated as the next one I'm going to show you. Shows the connections among the four science and engineering faculty who were involved in the acid mine drainage project. The four ovals towards the center, which I'm a little colorblind, they look a little purple to me, were the faculty members. And if you look at the ones on the upper part of that diagram, you'll see that, especially the person on the left, there's a lot of connection among the people in the group. If you look at the ones on the bottom, you'll see that the connections to the students, those are the other ovals, are really in a one-to-one situation with the faculty member, with the lead researcher. So the tightly structured has a lot of connections among the members of the group. The loosely structured, the members of the group are mainly connected through the lead researcher. Okay, now this next diagram is of a PIRE project from NSF, Partnerships in Research and Education. I can't remember what the name of the acronym stands for. But here you see a lot more connection, a lot more complicated type of grouping. If you delve into this, you'll see that some of them are still more tightly connected and some are more loosely connected than one another. But basically what you're seeing is that if you develop a good interdisciplinary work group with a lot of collaboration, you get these sorts of connections. Okay, so research groups are communities. Now, the language on communities tends to talk about communities of practice. And communities of practice are groups of people who are engaged in a particular practice. That practice could be doing research, that practice could be teaching, that practice could be doing medicine. But there's another way in which we can talk about communities, and that's as in epistemic communities. And epistemic communities are ones where not only are they engaged in some practice, the practice that they're engaged in leads to the development of new knowledge. So research groups are both communities of practice, people who engage in the actual methods of research, and are epistemic communities, because the ultimate goal of an epistemic community, ultimate goal of a research group, is to produce new knowledge. So then the participants in the research group think about it in terms of the peripheral legitimate participation, where the novice researcher is the person who's starting out in the group. The proficient technician has gained a lot of skills, and the knowledge producer is one that not only knows the methods of the research, but also is helping in significant ways to produce new knowledge. And those terms, by the way, came out of the language that the professors in the acid mine drainage research project used to describe their students. So I didn't invent those terms. They came out of the professors themselves. So the novice researchers, well, they're short-term members of the group. They could be, an undergraduate who's part of doing an undergraduate internship. It could be an REU. It could be a master's degree student who's very quickly doing a master's thesis. But typically when they start out, they lack research skills, and we use the term methodological proficiency to refer to a lot of those research skills. But they're skills that they don't have. They're not good at formulating research questions. They have difficulty coming up with defensible conclusions. They're not expected to contribute much of anything to the analysis of data or the creation of new knowledge. And those are skills that we classify under the category intellectual proficiency. And all of these categories are fuzzy. And they're fuzzy because what we're doing is we're describing people and what people do. And description of people and what people do cannot be put into clear-cut categories. They're always fuzzy. All right, so the proficient technicians, I might have skipped this out here. The novice researchers, so the proficient technicians are longer term in the group. Often these are the master's degree students. They come out with skills that enable them to engage in a lot of different types of research methods. They can write good reports about what they've studied, but they may not actually develop original research questions. They might get them handed to them. If they're working in industry, some proficient technicians are working in industry, they're given some tasks to do by their boss or their client. And so that was what the third bullet was. And then the knowledge producers are long-term members of the research groups. They have the methodological proficiency, they have the intellectual proficiency, and the scientists and engineers who were working with me on the acid mine drainage project said that's what they wanted their doctoral students to be when they completed their doctoral program. They wanted them to be knowledge producers who had all of these different types of skills. So, the expertise grows. It grows from novice researcher to proficient technician, from proficient technician to knowledge producer. And for all of this to happen, the students need to be, well, the way in which this typically happens is that the students are engaged as apprentices in the research groups. They're given different tasks that are manageable chunks that they learn to do, and those chunks are scaffolded to be more and more complicated. And over time, they get to the point where they go from knowledge novice researcher to proficient technician and eventually to knowledge producer. And we have this diagram that we developed that sort of shows that, that path. You can see that there's the research group has aspects of the community of practice, aspects of the epistemic community. The novice researcher, comes in, zooms out. The proficient technician comes in, spins around in the community of practice, might head into the epistemic community a little bit. And then the knowledge producer is spanning both of those aspects of the research group. So what does this mean for cures? Well, it means that you need to have realistic goals in terms of the methodological and intellectual proficiency, and for the knowledge and skills needed to participate in an epistemic community. So the goals for a cure have to be realistic for, this very short time period. If we go back to this, the cure is the equivalent of the amount of time that a novice researcher is spending in the research experience. The type of research group matters. We found that the tightly, that's the typo there, should be tightly structured, not tightly loosely. Tightly structured are more efficient than loosely structured because all the participants are interacting with each other in the research group, and they're all learning from one another. They're sharing knowledge, knowledge grows among them. And then if you're going to be doing this in the context of a course, you have to have, it has to be highly structured with well-defined research projects and scaffolding activities like journal clubs and regular research group meetings, and of course, assignments that could be part of the course that relate to that. So I designed this model. This is now a course-based model, not a theoretical model, on all of that. There was a 3-credit course on how to do research that provided the scaffolding. There were research experience in STEM faculty research groups and mentored by graduate students. The next couple of slides are details about the course components. I'm not going to go through this because it's too much to go through. There's too much on the slides, but you'll have copies of this. You see what that looked like, and what was supposed to be, and then how we used in the next example that I'm going to talk about. So we actually had 11 scientists and engineers, faculty members at USF, who agreed to open up their labs to students who were enrolled in this course. These are just a couple of examples of them, one from geosciences, one from environmental engineering, integrative biology. USF has 2 biology departments, cell and molecular and integrative biology, and then the physics department. So 11 faculty said, yeah, we'll find a space for these undergraduates to spend time in our labs. And this is a diagram of that model. So I don't know whether you can see my cursor, but this is the course in the center. There's a course instructor who facilitates the research and provides explicit instruction on how to do research. The students in the course are arranged in research groups. Each of those research groups has a graduate student mentor, and then is working with a research group from one of those faculty who I had in the last slide. Well, I put in a proposal to IUSE to fund this, and basically the reviews I got was, well, this is too ambitious to do in one semester. And I said, well, the idea was that this was an experiment. We're going to see what we can learn from doing it in one semester. And then when it came time to resubmit it, like I said, the pandemic hit, everything shut down, and we're just now regrouping and thinking about resubmitting it. So, but we did have the opportunity to do a pilot study. So the pilot study is a undergraduate science education course at our university that's called Science, Technology, and Society. The person who taught it for forever, and retired after a long career. And I said, all right, I'll teach it this academic year. I taught it in the fall, and I'll use it as an opportunity to introduce 2 things into it that were of interest to me. One is a focus on the food, energy, water nexus. My background is actually in physics. My original background is in physics, but over the years, I've moved into environmental issues, environmental science, and the food, energy, water nexus is an important way in which environmentalists are thinking about the ways in which human beings interact with the natural world. And then we could also provide students with the opportunity to engage in research activities that's related to different aspects of the Nexus. So in terms of science education, why it matters is that there are these next generation science standards that say that K-12 students need to learn how to engage in the practices of science and engineering. And if you look at the document that describes this, which is the framework for K-12 science, which the National Academy Press has published, you'll see that the goals for what this ought to look like by the time students graduate from 12th grade are very ambitious. They're really pushing towards that knowledge producer level. All right, but then on the other hand, the people who are teaching these students, the teachers, have never engaged in research. You know, very few undergraduates, that's why we're interested in cures, have ever engaged in science or engineering research. So how are they going to learn how to teach their students how to engage in research practices if they've never done it themselves? I mean, they don't have to be a Nobel Prize winning scientist. They should have had some time doing it. You know, it's sort of like the same way the coach doesn't have to have been on a World Series team, but they better know how to play baseball if they're going to coach a team. So we use this as an opportunity to do research on this class, we being myself and Rita Ortiz, who's going to be facilitating one of the breakout rooms. We asked, well, does this actually help them learn the practices of science and engineering? What effect does the FUE Nexus have on them? And will this have any effect on their future teaching? Of course, we don't have data about their future teaching, but we do have data about what they say they would do in the future. So the class was relatively small. There's only 11 students. One of them was not a science education major. Our biomedical sciences major is basically pre-med. For some reason, the biomedical program is in our chemistry department. That's called University Politics. Everybody was either junior or senior, 8 women, 3 men. We also had 2 graduate student mentors who helped with this, and was a very diverse student group, not only along racial and ethnic lines, but also in terms of age, experience, and goals in their lives. So we did some surveys. Now there's an instrument that I developed based on an existing survey on perceptions of abilities to engage in research skills. Clearly, we did observations of the course. Rita was a participant observer. I was the instructor. We had a variety of different assignments and activities that it did to scaffold the learning to do research. And then Rita interviewed the students. One of the things that we did because of the IRB, Institutional Review Board, Human Subjects, is that Rita was the one who asked the students whether they would consent to be part of this research. And she held on to the consent forms. And I had no knowledge about who said yes and who said no until after I submitted grades. So that was a way to reduce coercion. So there were 3 research experiences that we used that we had actually borrowed from ones that we had developed for the use with high school students. So the one on the left, and you see that the equipment is very, very simple. The one on the left was the growth of algae and the factors that affect the growth of algae related to red tide, which is a significant problem here in South Florida, or at least West Central Florida. We've done this project with the use of biosand filters to produce potable water. We did that with high school students in the Tampa area and also in Ghana. And we're now moving towards this use of studying of biodigesters to produce compost and biogas from food waste. So the biodigester equipment, if you can see that, this was in a student's garage. The bottles are actually from Perrier water. You don't need that fancy water. And then those collection jars and the dishes I got from the dollar store, from Dollar Tree. Here's a schematic of the biodigester project. And you can see it's very simple. We have the feedstock, which has to be from an ungulate, and some food waste, and then the plastic tubing. The gas tea and a clamp is a way to get samples of the gas to measure the amount of methane and carbon dioxide in the biogas. And this shows the collection through using a graduated cylinder, but in any sort of container could be used. Interestingly, the way that we got the cow, goat, and sheep manure for this is that not too far from the university, there's a halal slaughterhouse, actually, not just a butcher, that starts with the live animals, and then they kill them, and then they sell the meat. And since they had the live animals excrete. So that's where we got that from, or they got them from. I sent the students to go get that. So some of the things that could be done in the scaffolding is to look at some data and ask some questions about the data. So this was actually from the research lab that in the Department of Environmental Engineering, where they're working on biodigesters, but using very, very similar equipment, but a variety of different substrates, looking at the methane yield. This also shows what happens in terms over time. And then this third one looks at the effect of data, of temperature on the production of the biogas. So these three things could have been tested by my students. There was one group that focused on the biodigesters, and they looked primarily at the different substrate. But there's a wide variety of research questions that can be answered by any of these three projects. So what did we find? Well, our first research question was, what did they learn? And well, they did learn some methodological and intellectual proficiency, but mostly methodological. They really didn't progress beyond the role of novel researcher. That said, they did feel like they were working like scientists. They'd never experienced anything like this in any science course that they'd taken before, either in high school or in college. They wish we had focused more on providing them with a literature review. We feel that it would have been better to reduce the number of choices of the projects. One of the things they did was they went on a lab tour to see these activities being done in an actual laboratory setting. And the graduate students were helpful, but they themselves had very different levels of research expertise. I know I'm running out of time, and I only have a couple more slides, so I'll go through them quickly. So, not surprisingly, the focus on the food, energy, water nexus was highly motivating to them. They saw that what they were doing had real world connections, and had connections to real problems in the world that they could help solve. And they thought that they would be able to use the Nexus to motivate their own students when they were teachers. Oops. And then they basically all said that they would incorporate some type of authentic research experiences into their own teaching. In the final paper that they wrote, they described how they would do it with their students, and their descriptions were pretty thorough. But they did acknowledge that there were a lot of barriers that they would need to face, for example, the coverage for high-stake exams. So we have some references on the PowerPoint that you'll get a copy of. And other than that, I guess we'll just talk about this in the breakout groups. So thank you.
Great. Thank you so much, Dr. Feldman. We look forward to continuing the discussion in the breakout rooms. But I'd now like to turn it over to our second presenter, Dr. Lena Dahlberg, who is an associate professor of biology at Western Washington University. So Lena, over to you.
All right, thank you so much. Thanks to everybody who has shown up today. So I'm going to be talking about collaboration in course-based research experiences. And just by way of introduction, I'm coming to you from Western Washington University, which is about as far from Florida as you can get. We're all the way up here near Canada. We're a primarily undergraduate institution, but we have a lot of students, so about 15,000 students, and 85% of those students are from Washington State. I personally am a split personality, so I have a cellular biology research lab where I study ER-associated degradation. And then when I think about organismal biology, I think on the student level. And here's where my biology education research comes into play, and that is as someone who's really interested in course-based undergraduate research experiences, or CURES, how they can help educate student researchers, how they can introduce faculty-driven research into laboratory classrooms. And I use this model as a way to understand how students approach problem solving. So for my talk, I'll get to an outline in a second, but I'll give you the punchline at the beginning, which is that I feel very strongly that by focusing on elements that students value when they're learning, and as part of their learning, we can strengthen laboratory courses, even if they are not full-blown course-based undergraduate research experiences. And as science educators, I think it's really important that we think about how we can be building communities of people who not only are interested in answers for their own sake, but who are interested in helping each other search for answers to real and important questions, such as the one that Dr. Feldman just described. So I'm going to start out by talking about some important elements of CURES, and also, as Dr. Feldman just pointed out, some of these are wishy-washy, but I'll use them as a framework. And then I'm going to talk about ways that you can and we can include meaningful collaboration in laboratory courses and CURES or CURE modules, so partial courses. And then I'll end by giving some examples of how we have introduced collaboration at 3 different levels in our coursework here at Western Washington University. And I'll end by turning us over to discussion breakout groups that I'll introduce again at the end, but essentially thinking about how you might bring collaboration into your own coursework, and then also thinking about how you might expand CURES to make them more collaborative in the research topics. So really briefly, there are many ways to run a CURE, and as a biologist, I'm very interested in how biology CURES are run, and I'm highlighting here at least 3 examples, and I know there are more. We can see single courses that are driven based on faculty research, linked courses where a number of courses come together to form a larger project. And then there are some great examples of dispersed programs that ask big questions. For example, the Tiny Earth Project looking for antibiotics in soil-dwelling organisms, and the Genomics Education Partnership that's been very successful and has a whole training project associated with it. So there are many ways to run CURES, and I just want to put that out front. But one of the main things that's so great about CURES is the fact that they can provide students with these laboratory research experiences, which gives students hands-on time with real research. They can require students to assess, plan, evaluate, monitor, and reflect, which is sort of a long-winded way of saying they induce metacognition in students. They promote science identities and scientific confidence, and they're able to reach larger and more diverse groups of students than independent research opportunities in, say, a faculty laboratory. So course-based or CURES can provide students with this authentic laboratory setting in which to learn, and they include such highlights as being able to do scientific practices, discover new things, do broadly relevant or important work, collaborate, and iterate, so repeat meaningfully. But putting all of this into one cure can be time consuming and expensive, and having a course that is just based on a single research question, for example, or several research questions investigated by students can be time consuming and very expensive to put together. Dr. Feldman also alluded to that. So my research group took on a sort of different tack, and instead of saying these are the things that must be in a cure, we asked what elements of course-based research are the most important for our students and why are they so important? And to answer this question, we introduced course-based research across several levels of a biology curriculum. We then identified elements that students reported as important. We interviewed students to discover how those elements impacted student learning. And then we've been trying since then to revise courses to include and improve those elements, in particular with a focus of looking at what was important to students as they were going through those courses. So I'll start out by just showing you what this course-based research project looked like at a departmental level. Western Washington is on the quarter system. So we I'm showing you here that we have an introductory series that runs 3 quarters a year, a mid-level course that runs several times a year, so up to 4, and then advanced level courses that are variable. And they serve different numbers of students. And we were lucky enough to be able to provide authentic research modules or full-length courses in a number of different iterations of these courses, which I'm showing you by the darker colored boxes here. And that's over several years now. And we focused on faculty-driven research projects in our department that could be introduced into courses. So using model systems or place-based research questions. There's an exciting arrow here that shows up that there was in fact a pandemic, but we kept on running the courses. In particular, we were able to continue running the research courses at the advanced levels, which was I think a great feat and really important for our students. Based on the interviews and the assessments that we gathered from students who had gone through these courses, we found that 2 elements of cures were really, really valued by students, and those were collaboration and iteration. And we've published on the iteration portion, and you can see those references at the bottom here. So today I'm going to talk to you about this idea of collaboration and how important it is for student learning as they're going through these sorts of courses. And particularly, I want to talk about ways that you can include meaningful collaboration in your laboratory courses, whether or not these are cures or cure-like courses, or whether they're more like inquiry-driven or even sort of traditional laboratory courses. And specifically, we think it's important that these can be cheap and easy ways to include elements that we see as important in cures that can be introduced in a number of different courses. So the information that I'm going to show you in the next 4 slides are included also in a handout that you can get from the website. So I won't spend too much time reading all of the text, but I'll go over what the big ideas are. Essentially, what we did was we took these ideas that students were giving and reporting back to us year after year, and we compiled them into a set of guidelines that we think are useful for thinking about how you can include collaboration as part of course design. And they introduce collaboration both as they refer to research in the real world or in a laboratory classroom, but also as it applies to the student experience as they're going through the learning process. So what I'm showing you here is our first one, which was we found that low stakes collaborative work or lab meetings are really, really valuable to students. And so here's a picture of students who are having a non-graded time to communicate their understanding and their discoveries by writing models on the board. And the goal here was simply to practice science communication and to understand that people were having the same kinds of troubles or confusions between individuals. On the right here, you can see an intermediate level lab where students were having trouble with a technique. And by bringing them together as a lab meeting, they were able to understand what sort of common mistakes were being made. We found that students valued being given time to struggle and to struggle as groups, because it allows them to understand how long it can take to learn something, and also to find out who and where their trusted sources are, which is something that real researchers also need to learn. And what we also found was that students really valued being given the time to build that understanding. And so it's not about efficiency when students are learning struggle. It's about letting them have the time to struggle as a group together. We found that students valued interacting with science communities on campus. So being able to understand where samples were coming from, for example, in one set of students, those samples then being assigned to different students who maybe processed the samples and then sent them on further for further analysis, was really important for students to understand the idea of how to have confidence in a collaborator and understand why that makes a collaboration helpful. But it also helped students build trust between themselves and between classes and different science communities on campus. And science communities off campus can be strong collaborative sources also, where students who get to interact with large research universities, we're a small research university, large research universities with large labs, getting to see that the folks that they are working with are using the same techniques and having the same troubleshooting problems was really valuable to students, to understand that they could be part of the troubleshooting process because they weren't alone in learning the steps and discussing the problems they were having. So along with that, at the bottom part of the worksheet, or the handout that I've given, we've suggested some ideas about how collaboration can be progressively redesigned into here or other laboratory courses. First, by, for example, providing a time for students to share their results, but over time, perhaps revising so that the grading becomes less important for students, whereas the process of doing collaboration becomes more important, leading to a possibility for students to see their peers as valuable and irreplaceable resources in the research that they're doing. And this is just one example, but I'm happy to talk in the discussion group about other ways that that collaboration can be progressively redesigned into CURE courses. So that's a really brief overview of some of the findings that we had. But what I'd like to talk about for the last part of my talk is this idea that there are lots of different models for how you could introduce collaboration at the course level. So I'm going to give 3 examples. One is temporal, so collaboration over time. One is intramural, which is collaboration across campus, and one is extramural. So temporally, what we found was that because we're working across academic sessions, we could introduce a scientific question early. And so here what we have are students who are genotyping a novel strain of C. elegans, and then later draw on the research that was done by a previous course to gather new data. So these students had actually, the students who were in the advanced lab here had never met the students who did the genotyping to make the animals that were available for their research project. And so temporally, these students were trusting each other over time to get their, to bring their project to fruition. So we've done this several times now, and students can take and trust the results of previous students, which is really, really fun for them. Intramurally, we've had great success working across the building or across campus, and we've done most of this work using our advanced laboratories, and these are 10-week, 1-quarter courses. And what we found is that the collaboration between advanced laboratory courses can expand the range of research questions and available techniques that students can use. So in one example here, we have a biology course and a psychology or behavioral neuroscience course where students are actually providing each other the reagents that they each need to continue their research project. And along the way, we provided joint group meetings so that students could describe the methodologies they were using, as well as the troubleshooting that they had to do or were continuing to do. So each of these courses were independent. The students had final products that they were turning in, but the end result, the end goal was that these research students would be collaborating towards a manuscript for publication. Another example we have of an intramural collaboration are two different biology courses where the biology question was linked to two faculty-driven labs, but each of these courses run sort of sequentially to each other. We're providing strains and materials for the next lab. And in this case, again, we had individual projects coming out of each course, but the data that students gathered was actually of high enough quality to be used for successful grant proposals, which I think was also extremely important for the students to understand. And so what I'm showing you here is the range of data that students can use when they're allowed to sort of cross-pollinate between courses. So we would not have been able to do all of these different techniques had each of these courses run only by themselves. But by bringing courses together and allowing them to collaborate, we end up with a more realistic research project that looks more like something that would be publishable in one of the larger journals, scientific journals. So I'm going to end by talking about extramural collaborations. And the extramural collaborations have been really fun to set up because what they're doing is they're providing a way for students to expand their horizons of how research is done and who does research. And so on the left here, what I'm showing you is a collaboration we had with the University of British Columbia, where there was a large-scale genotyping project happening. And we partnered with the Mormon lab at UBC to send us strains of animals that had not previously been fully genotyped. And we asked our students to do that genotyping. The students then, using the understanding that they had about PCR, about running gels, and about troubleshooting, then wrote up a little lab report that we would send back to the collaborating lab as a set of data for them to work off of in fully genotyping the animals that they were using. And this has been really successful. We ran this for I think 4 or 5 academic quarters. And this is an example of, again, where this extramural collaboration also turned into a temporal collaboration, where as students were able to work with the worms in one quarter, 2 or 3 quarters down the line, we'd be ready to use those animals as part of a different experiment. Obviously, this works really well when you're in a lab and you can be there in person. We ran into some hiccups with the pandemic. And so one of the things that I thought about as the pandemic hit was, how could I help my students understand that science kept going even when there was a global pandemic and we were stuck at home? How could we get that into the classroom? And what I did was I set up a partnership with Wormbase. So I work with C. elegans, and the C. elegans community has a database that is very wide ranging, but one of the great things that they have going for them is a way for researchers to annotate previously published work. So I partnered with Wormbase and asked my students to become part of the scientific community outside of our university and annotate data that they were using in their own literature-based projects. And the outcome of this was really fun because it turned out that through me, so this is, I go by Lina, but Caroline, that's me. Through this, my students actually became the second highest group of community curators in a 3-month period. But even more exciting was the student here at the bottom who took on his own worm person ID number and made it into the top 10 community curators on his own. And the students reported back that not only had they learned an awful lot about reading primary literature and understanding what a control meant, but they really felt like they had become part of the scientific community because they were providing something to the public sphere. So I went through it quickly, and I'm happy to go back with slides, but I otherwise think that going through the going out to the breakout groups might be the most productive way for us to talk more deeply about all of these topics. But what I want people to take home from this is that designing and implementing a full cure can be time consuming, it can be expensive, and it can be expensive to the point of being prohibitive. But by focusing on elements that students value as part of their learning, we can strengthen laboratory courses even if they don't become full cures. And collaboration is an important part of research that students value, but they don't always learn that it's really an important part of everyday professional science. Collaboration can be easy and cheap to include in course design because simply putting aside 30 minutes for a group meeting can really change the way that students think about their own research project. And Dr. Feldman was talking about how students are able to communicate or not in their own project. And having these collaborative moments can really help students gain in that. And then collaborative research courses can lead to productive and relevant student research experiences, just like collaboration in the professional science world leads to more productive and relevant research. So I'm really excited to spend some more time studying how collaboration helps students learn. But in the absence of that data, I think that what we've seen is that when we focus on what students love about learning in the laboratory course, we can strengthen those courses. And again, I said this is the same slide as at the beginning. As science educators, it's important that we're building communities of scientists, not individual scientists. And so this idea of putting collaboration into coursework can be a really important way for students to start recognizing that as they're learning how to do science, part of that is learning how to speak to each other, learn from each other, and trust each other. So with that, I will thank the NSF for the funding they provided for this project. And it's a little hard to see, but Jackie Rose, Nick Galati, and Ben Wiggins are all here to help out with the breakout groups and discuss their experiences. Jackie and Nick will talk about their collaborative cures. And then Ben and I will be available for talking about how we can introduce and monitor how collaboration is working in our coursework. So thanks so much for your attention, and I look forward to the breakout groups.
Welcome back to the main session, everyone. I'm wondering if we can get our facilitators and speakers to share a quick recap of what their discussion group talked about. So I'm just going to go in the order of groups. So I'm going to turn it over to Alan first.
Okay, so first I have to say that I didn't follow directions and I didn't take notes. So I'm just going to have to do it based on what I remember. I think the discussion really wasn't that much different from what I was expecting it to be. And that was really, how do you do this? How do you make this happen in my situation? Or how do you make it happen in this other situation? And one of the things that happened was that there was a sharing of the different situations that people were in. There was some discussion about, well, what really counts as research? You know, one of the participants in the group Bill was talking about how he has his students do this simple activity where they're growing mung beans and they have a control, and then the variable is that they introduce nanoparticles into the growing environment of the mung beans, and then they keep track of the growth. Is that research or not? And so we talked about that. One of our participants, who was the in charge of, well, I shouldn't, maybe not in charge, but the director of undergraduate research at my institution in the University of South Florida, suggested that, you know, you could look at some of the sustainability goals. I think the UN has something like 17 of those. That could be the focus of different types of research experiences for an interdisciplinary research course like Hassan was describing. So that was basically what we talked about. I mean, the other participants might have other things to say.
Great, thank you. I'll turn it over to your colleague in one of the other breakout discussions. So Rita, would you like to share a quick recap from your group?
Yes, hello everyone. So we first looked over some of the data collection, that part, and we looked over some of the findings, and we talked about what research roles did the groups or the research project groups have. We talked a little bit about the design, the experimental setup, and whether the students had the choice on how to create, for example, these biosand filters or biodigesters. What did that look like for them? Or if they didn't have that opportunity, and why. We also talked a little bit about whether it would have made a difference if the students had the chance to look at previous research about these projects, and they can kind of see what worked with their projects and how they could tweak it. We also talked a little bit about the engagement of the science and engineering practices, whether they had the opportunity to present their findings. And we also talked about what this would look like in differently other than biology curriculum. I believe that's pretty much it.
Great, thanks so much. Thank you to both of you for leading those discussions. I'll turn it over to the other presenter group. So I'll start with Lina.
Great. So yeah, our group had a pretty wide-ranging discussion. We talked about collaboration from a variety of different levels. We talked about how to assign authorship when you have a collaborative project. And we talked about what happens when you get a very large group of undergraduate authors. How do you define intellectual, contributions to a paper. We also talked a lot about how to capture student values. So I talked a little bit about the techniques that we had used, looking at student responses in the interviews and also in written work, and basically our coding process and how we came up with defining collaboration, and also repetition is really important to student learning. And then we talked a little bit about what do students want, what do students want in courses, and how can we think about the idea of taking grades away from something that's so important like collaboration, while it's still at the same time keeping students engaged and also accountable for the work that they're doing. So we talked a little bit about grading for there being a presentation, maybe not for the content of the presentation so much. And we talked very briefly about the idea that that students in different disciplines might have different needs, and I don't have a lot of experience with that, so I didn't have a great answer, but I think that it's something that's really important to think about, is that students who are coming from a field other than biology might have different perspectives. Like science culture is really different between fields, so learning that science culture for the students could also be really important.
Awesome, sounds like a great discussion. I'll turn it over to your colleague, Ben.
Awesome, yeah. I'll second the interesting mix of different fields of science that were coming into our discussion. I think we took a pretty pragmatic angle on just how do you make collaboration work? So are you giving cues at the beginning of the process for how you want people to collaborate? Or are people bringing their own ideas of collaboration to the room, and then you're adjusting that with feedback somehow, or with getting students to reflect on the experience, and then how making that a meaningful part of the experience. My group members, feel free to jump in, but it really seemed like the tools that we choose to adjust how a group moves, or how a group of groups move, they're going to vary institution to institution, class by class, but they're all going to take into account a sense of this value that the instructor is hopefully pulling up from the class itself, but then sharing out as this is how it's done in the profession. And so we had a lot of really good ideas about specifically, is this a piece of paper? Is this an assignment? Like, how do you make that happen as the tinkerer in all these groups?
Great, thanks so much. And finally, I'll turn it over to Jackie.
I'll just be brief because most of what we discussed has already been mentioned and we're over time, but really the logistics about collaborating across courses, timing in terms of if you're in the same quarter or across quarters, and also assessment was brought up, like how do you assess both an individual in terms of what they've learned in your course compared to if it's more of like a group project because of the collaboration, how does that assessment go? And then also mentioning like what types of assessment are of interest to external agencies too for that kind of experience. But if anyone from my group wants to jump in as well, please do, but I'll leave it at that for now.
Great. Well, I just want to close today's workshop by thanking our speakers, Dr. Feldman and Dr. Dahlberg, as well as our breakout facilitators, Rita, Ben, and Jackie. Thank you so much for being here. I'd also like to thank the rest of the AAAS-IUSE team and all of you who were able to attend today's workshop. We do have a post-event survey, which is sent out by email. So we really appreciate your feedback. It should only take a few minutes, and we'd really like to continue to improve these workshops in the months to come. So once again, thank you all for joining us today. And as always, we look forward to continuing to engage you and learn from you, our AAAS-IUSE community. Have a great rest of your afternoon, everyone.